central dogma in molecular biology. We know DNA is a genetic material which stores the information in the form of letters like nucleotides. These letters are adenine, thymine, cytosine, and guanine. That is A, T, G, and C. And these letters, these letters actually is the storehouse of informations within the cell. And DNA is present in the nucleus, which is inserted and in a compact form in the form of chromosomes. And central dogma of molecular biology explains a unidirectional flow of genetic information from DNA to RNA and from there to formation of a functional proteins. And this concept of central dogma was put forward by Francis Crick in 1958. In this image, you can see the flow of information that is from DNA, the double-stranded DNA, to RNA, which become a single-stranded, and from the it forms a functional protein. And hence, the DNA will store all the information necessary for synthesis of all the proteins or enzymes which is needed for the metabolic activities in our body. And here, the RNA acts as a messenger which carries the information from DNA to become a functional protein. And this synthesis of protein occurs in ribosomes, which are the factories which helps in protein synthesis. The information from DNA is read in RNA in the form of a genetic code, which are the triplet codons. And each triplet codon codes for an amino acids, which forms the basic unit of proteins. And in this image, you can see a start codon AUG, which is a triplet codon which codes for an amino acid methionine. Similarly, this is a strand of mRNA where the start codon starts the synthesis of protein followed by triplet codes. These three are the different triplet codes. Similarly, there are several triplet codes are present which codes for an amino acid. Now, the central dogma is broadly classified into two, mainly the transcription process as well as translation process. And reverse transcription is a rare event which occurs in few viruses. And in this image, you can see the transcription occurring from DNA to RNA. That is the transfer of information from DNA to RNA. And translation means it is the transfer of information from RNA for the synthesis of protein. And in transcription, the information in the DNA in every cell is converted into a small portable RNA messages by the enzyme RNA polymerase. And so we can simply say that transcription is the transfer of information from DNA to RNA. And in this image, you can see the blue color is the RNA polymerase enzyme, and which helps in the transcription of double-stranded DNA and form a new mRNA. So, next part of central dogma is translation. And during translation, the messages from DNA in the cell nucleus is received by the RNA and it is read by the ribosomes for the synthesis of protein. And so we can say that translation is actually the transfer of information from RNA to form a functional protein. And in this image, you can see the ribosomes which are in blue in color, and a ribosome reads the information which is present in the mRNA. This is the mRNA strand. And as part of the reading of the information, new molecules of amino acids which forms the basis of the proteins are formed. Now, next is a reverse transcription. It is a rare case which is occur especially in certain viruses, especially that retroviruses. Example is HIV. HIV is a retrovirus where instead of DNA, RNA is the genetic material. In that case, the genetic information which is stored in the RNA is converted into DNA. In normal case, the DNA is transcribed to form RNA, but in reverse transcription, opposite of it occurs. That means RNA, which is genetic material, will transcribe to form DNA. That is referred to as reverse transcription. And this process is initiated with the help of enzyme RNA-dependent DNA polymerase. It's also known as reverse transcriptase. So we can conclude that the central dogma includes both transcription and translation. That is, the flow of information is unidirectional from DNA, then to RNA, and from there to protein. So the ultimate product of the DNA synthesis or DNA expression is protein. Thank you.